Okay, so today what I will be talking is about fertilization. So I was supposed to do oogenesis before, but I think this is a more important topic. And for since it's a large topic, today I will not be writing because it will be taking a lot of time. I'll be explaining my notes. I'll be explaining the notes. Then that will be easier in some sense for me as it should not make a problem for you because you will be studying that note. So, for developmental biology, I would recommend Gilbert. You will get this book online. If you don't have, I will send you the PDF. Try to read from that book. Don't have to buy those books. Or on if you have a PDF version, try to get accustomed reading them on digitally. You save paper actually. You save paper if you read them. If you make a printout, then it's the same thing. It's cheaper, but you waste the paper because you read it for a few days and then forget. Better to keep everything digitalized. Even in former times, I used to make printouts of my notes and give it to students. Like in for earlier times, I used to have this big, fat all note collections. I used to make them. Students used to pay for them, of course, and then I used to distribute those notes to everybody. But then I saw that people, it was not working well for the environment, for everything, and cost wise also nowadays it's not effective. Former times it was 50 paisa per page, but nowadays they're charging 2 rupees for printing in Calcutta, and then it's not cost effective. For these notes, it's not cost effective. For the book, it was okay. So best is try to read them digitally. And what I always suggest is all the notes which you get digitally. If you have problems in reading from a small phone, try to write it down in your handwriting. Copy it. While you are copying it, your preparation will be over. At least the initial preparation is over. This is what I used to tell my students. But unfortunately, at that time, I was very dependent on my tuition. My salary was not very high. So uh, I used to sort of whatever they want, I used to give. But then I used to teach them. And anyway, but then I realized that I was not doing a good thing. So because then I think it's for last 1 or 2 years, what I was before that, 2 3 years, just before the COVID pandemic, I started digitally. And after this COVID pandemic, when it happened, during this COVID pandemic and after, people got used to studying things digitally. অভ্যেস হয়ে গেছে. So it should work well like that. Okay. So I'll share my screen. Your entire screen. I try to increase the size. Okay. So about fertilization, it is the fusion of haploid gametes, egg and oh, it's the screen is blank, na? So about fertilization, it is the fusion of haploid gametes, egg and sperm to form the diploid zygote. So before everything, let us have a overview of what are the things which happen. The first step is the binding of the sperm to the zona pellucida. The binding of the sperm to the zona pellucida. Zona pellucida is this is the thing, binding of the sperm, outer covering of the egg, that is the zona pellucida. Second, what happens, you have the sperm has got a tip known as acrosome, has got enzymes. The acrosomal reaction takes place. Second thing, you see it has bound here, this circle which you see over here. the sperm has bound to the zona pellucida, the outer covering. Acrosomal reactions take place and the sperm is dissolving the membrane over there. And then third, it has entered into the zona pellucida. Then the plasma, it is the fusion of the plasma membrane. The plasma membrane of the sperm fuses with the egg plasma membrane. জায়গাটা ফাঁকা হয়ে গেল, entry channel হয়ে গেল. And then entry of the sperm nucleus into the cytoplasm. It enters inside it. These are the steps. And then the next step is that it fuses. Now, if you have a closer look at the binding of the sperm, to the zona pellucida, you will see that on the zona pellucida, there are receptors known as Z3 receptors, these brown Z3 receptors. There are also Z1, Z2, but Z3 is the most important. So this is the acrosomal vesicle of the sperm, which you see on the top. It has got important, the formation of acrosome, it has got this thing. So it binds. This binding is a very important stage. A lot of reaction, chemical changes takes place in the egg. So where does this fertilization takes place? It takes place in the fallopian tubes. Now, what are the fallopian tubes? If you look at the female reproductive system, you will find this is the uterus, say for example. Then you have the cervix where they enter. Then you have the vagina, and this is the uterus. It's got the walls are known as endometrium, where the zygote gets an implant, and from here you have these ovaries coming out. But from that ovary there are this is the fimbriae. You have certain tubes coming up like this. It enters. আরেকটু সাইডে থাকে এদিকটা আমি লিখেছি. You have certain things like this. So the egg, after ovulation, it enters into these tubes, and the sperm comes in from here, and fertilization takes place inside these tubes. These tubes are known as fallopian tubes. These tubes are the fallopian tubes. These are the fallopian tubes. The site is the fallopian tubes. The passageway from the ovaries to the known as oviduct. In female mammals, this passageway is known as uterine tube or fallopian tube. So in vertebrates, reptiles, so for females, not mammals, you will see that they are known as oviducts. I don't know if you people have read about Mullerian ducts and Wolffian ducts. Basically, it's these things. So if every month, let us look at this picture. So, I've already have this picture. So these ovaries are over here. This is the ovary. Abharuchi, can you see the ovaries here? Hello?
Yes, sir.
Okay, yes,
sir.
Okay. So stages of oogenesis takes place, ovulation takes place, and the egg is released. I will explain this oogenesis. It's a process that's all, that the egg is formed. So this zygote goes over here and sperms come over here. So inside this tube, you can see in this region, there are sperms coming. And they have magnified this image over here. You see this particular fertilization which takes place, dissolving zona pellucida, it takes place in this region. If there is no sperm at that time, it happens they have not had sex, so the sperm is not there, and so the sperm egg comes over here and it degenerates. 2-3 দিনের মধ্যে নষ্ট হয়ে যায়. But the whole cycle will still be there. It will go till the next ovulation takes place, the menstrual cycle will start, periods will start, uh, because there is no sperm, no fertilization. But if there is a sperm at that time, fertilization will take place, and this fertilized egg in the stage of blastocyst. So you have blastula, morula, gastrula, blastula, and it comes to a stage where there is this whole little space over here. This is this blastocyst. It will come and it will implant in the uterus. At that time, this uterus becomes the walls of the uterine wall becomes very thick, richly vasculated, because it gives nutrient to this particular blastocyst or the egg, which has fertilized egg, which has come, and they start to grow. Then the placenta forms gradually from here. So this process is known as implantation. So if you look at the morphology of it, contains a single layer of non-ciliated and ciliated epithelial tissues. Oviduct is this fallopian tube. It's got one single layer of ciliated and non-ciliated. Ciliated means it has got cilia. একটাকে সরানোর জন্য দরকার হয়. And non-ciliated epithelium, it secretes mucus and stuff like that, which help, it lubricates that passage for easy passage. There is presence of smooth muscle that is essential for muscular contraction. This process is very important for fertilization. Many a times, many women during their early stages of life, they may get infected by this fallopian tube tuberculosis. It is not lethal, but you'll have fever and all these things. Before it goes to your lungs, it can go to your tubes also. And ovaries, this mycobacterium, it starts to damage these things. The smoothness, ciliation becomes fibroid over there, and as a result, neither can the egg pass out very freely, or nor can the sperms come in. So result is that they'll have problems having children. So in this case, it is always advised that they go through this process known as IVF, in vitro fertilization. They bypass this fallopian tube. They take the egg outside from the ovary through an injection. It's an operation, mild surgery. They take this egg out. They'll fertilize. They will fertilize it with the good quality sperms. They will clean up the total sperms, the good quality sperms, and then they will fertilize it inside a test tube or Petri plate. And the fertilized egg will be allowed to grow for up to 1-2 days. And then they will transfer it back into the uterus. The uterus will be made thick in this time by giving injections, which will hormones, progesterone and all, and they'll become thick. And these implanted things, once they are injected inside, these eggs are injected inside, they'll go and stick. And you'll have the pregnancy. Normally, depending upon the age, this is very successful. The older you are, the more chances are reduced. You have a 40% chance of success. 60% it will not bind. So pre-fertilization event, first thing is the passage of sperms. Sperms move up to 2 to 3 millimeter per minute using their own power. Power from the tail, the flagella in the tail, that will power. They must be helped along somehow. At the shortest, sperms can be found in the oviduct only 5 minutes after ejaculation. Some sperms take 45 minutes. So once ejaculation happens in the vagina, from the vagina it comes to uterus, from the uterus it will go to the fallopian tubes. It takes at least 5 minutes. So, but there is help needed. First is helping the sperm. So at the time of ovulation, ovulation means when the ovary is releasing the egg into the tube. A follicle is formed. Inside this follicle, the egg is developing, and then when it is released, ovulation, that is at the 14th day of the menstrual cycle. 13th to 14th day, this ovulation takes place, and that is the time when the female is most receptive to sperms. Chances of pregnancy is the highest. Those who are avoiding, they avoid this particular time, avoiding children. So, but still, it doesn't work. At times, many times, the previous cycle, some eggs are. So, all those things are different issues. So, other sperms can take 45 minutes, depending upon the strength and the quality of the sperm. And this happens due to the muscular contractions of uterus and oviduct. Contract করে, the sperms টাকে ঠেলতে থাকে উপরে. It pushes the sperms inside the oviduct. Then you have in the oocyte what happens. This is the ovary, and the ends of this oviduct, this fimbriae-like structures which you have studied. The oviduct starts from here like this, or I'll call it fallopian tube. They become very close contact during ovulation. একদম প্রায় ছুঁয়ে দেয়. The fimbriae, these are the fimbriae. The fimbriae of oviduct sweep the ovulated ovum into the oviduct. এখান থেকে যেই ফেটে বেরিয়ে আসে তাকে ওভিডাক্টের ভেতরে ঢুকিয়ে দেয়. It just pushes them inside. Once it comes out, the egg comes out, it is just pushed inside. And peristaltic waves মানে ঢেউ খেলে যাচ্ছে ধানের খেতে, like a wave, peristaltic waves, but one after the other, it's moving. It brings the ovum into the ampulla of the ovary, মানে ভেতরে নিয়ে আসে. Next, in the sperm, a process takes place, then that is known as capacitation, readying the sperm. Actually, immediately after ejaculation, the sperm has got a glycoprotein coat for the protection. It is just still not fertilization ready. A process takes place known as capacitation, and this glycoprotein coat is removed, and then the sperm becomes hypermotile. It moves very fast. This is this whole thing is written over here. So the sperms cannot fertilize oocytes when they are newly ejaculated. They need to be given time for 5 to 7 hours for capacitation. Sperms live in for 2-3 days. They can live in the oviduct, but the eggs after ovulation within 5-6 hours, they are not very receptive. They need actually the concept is that the sperms must be there. The ovulation they are there for 2-3 days. Egg comes and meets the sperms. It's not so easy. When you're wanting pregnancy, it will not happen. When you're not wanting pregnancy, lege jay. So how to make life tough? Like load shedding holo? Okay. Wait a minute. কারেন্টটা আজকে আবার হাওয়া বের হচ্ছে। Just hold on. কারেন্ট এসে গেল? Fantastic. So Ruchi, Abha, are you still there?
Yes, sir. Yes, sir. Yes.
Okay, they're there. There was a power cut. So capacitated sperms are more active. So location, it occurs in the uterus and oviduct and is facilitated by substances of the female genital tract. So the acrosomal reaction, where the acrosome dissolves the vitelline membrane of the egg and it enters, it cannot occur until capacitation has occurred. The uterus aids in the step by secreting albumin, lipoproteins, proteolytic and glycocytic enzymes such as heparin. কোনো clot না থাকে. And if heparin is there, then there is no clots formed. And you need it because one clot can destroy implantation. খুলে যায়, একটা ক্লট এসে গেলেই পাট করে খুলে যেতে পারে, মরে যেতে পারে। আমাকেও হেপারিন দেয় ফর আদার রিজনস। হেপারিন না, মানে আমাকে ব্লাড থিনার দেয়, দ্যাট ইজ ফর ইফ দ্য ব্লাড, মানে হার্টকে কার্ডিয়াক প্রোটেকশন, ব্লাড থিনার বলে, প্ল্যাটলেটসকে আটকে দেয়, যাতে মানে কোনো ক্লটস আর নট, দিস ক্লটস আর ওয়ান অফ দ্য রিজনস ওয়্যার you get heart attacks, myocardial infarctions. So even if things are accumulating in my arteries, in my heart, jomte thake, still, even if there is thing, this thin blood can move it. If you are aware of it, you can prevent attacks. Since I am a diabetic, the doctor has given, and I am above 40, so doctors have taken a precautionary and have given me blood thinners. চিন্তার নয়, ঝামেলা এমনি কিছু টেনশনের নয়, হট করে কিছু না, কিন্তু প্রবলেমটা হলে তখন মানে খুবই কষ্ট হয়। আর সেকেন্ড থিং হচ্ছে যে you have to go through angioplasty or stent or bypass in the worst cases. You have to bypass all these arterial block. There are three main arteries which go to your heart. Similarly, these clots are not good. So the process of fertilization following 5 steps. One is chemoattraction. It is said that sperms smell their way to the eggs. Smell. That is they have got chemotactic receptors which detect, okay, there is a gradient of a chemical over there. It's almost like smell. So when I was a student, I would say this was 1993, 94, 95, something around, I don't remember the time. At that time, there was no internet, so we used to go to British Council. So in Nature, I found this article, sperms smell their way to the egg. From there, I learned that there is something that was new at that time. This process of chemotaxis, it was new. Now, it's coming into the textbooks. In my time, it was not in the textbooks. We did not read it. So I read about it and I included it, and still I got second division. Those days were different days. Nowadays, without giving exams, you people get marks, and those are times, even if you give the latest and the all new information, they will not give you. Release of acrosomal enzymes. The next step. Then binding of the sperms to the extracellular envelope of the egg. Then passage, you have seen these steps, passage to the extracellular envelope. Then the sperm and egg pronuclei, the nuclei will fuse to form the zygote's nuclei. So sperm chemotaxis, this ligand binds this particular signal transduction, it takes place on the membrane. This is just something you need to practice. It's just like this ligand will bind to a receptor. this GC receptor. This will cause GTP to become cyclic GMP. Guanosine triphosphate will become cyclic GMP. The increase in GMP will cause this particular potassium channel to release potassium outside. Potassium will go outside. This will cause hyperpolarization. So you know membranes have got negative charge outside, positive inside. Hyperpolarization will change. Outside becomes positive, inside will become negative. Hyperpolarization. And this hyperpolarization will result in the sodium, calcium, and binding of cyclic AMP will result in sodium, calcium entering. After this, after a hyperpolarization comes the depolarization, where it becomes back into the negative state of the membrane will be restored. উল্টোটা হয় nerve impulse এর মতো, and calcium will enter and the flagella will start to move then. So this ligand is the chemoattractant. When the ligand from the uterus will bind on the receptor of the sperm, immediately ওর ল্যাজ নাড়তে শুরু করবে. It will start wagging its tail. You give some food to a dog, the dog will start wagging its tail. So the smell of the food, it will also start wagging its tail. আবার গন্ধতে এই গন্ধটা যেই হলে ল্যাজ নাড়বে, ওই গন্ধের দিকে এগোবে. It's being pulled, so it's almost like it is forced. যেই স্মেল এলো ওদিকে উড়ে চলে গেল. So this is the chemoattractant. It is secreted by the oocyte. অনেকে বলে the identity is not known, but one of the secreted is the steroid progesterone. Progesterone does something like this. Other ANP, atrial natriuretic peptide, ANP, it's also one of these sperm chemoattractants. But exactly you don't know. These results, studies have been done in sea urchins. So there is a chemoreception which takes place. The second step is the release of acrosomal enzymes or acrosomal reaction which takes place. The zona pellucida ZP3 glycoprotein, which you had seen, ZP3 is important in binding in the first picture. So the prerequisite, it occurs when sperm come in contact with the corona radiata of the oocyte. Then perforations, sperm and the corona radiata is, if this is the egg, outer vitelline membrane of the egg, some follicular cells are present. A region jelly-like, the cellular, something like this is the outside, like the sun, corona of the sun, corona radiates outside, something like that. So once the sperm comes in contact like this, this acrosomal reaction starts to take place. It has got ZP3 is over here. Some perforations on the sperm acrosome. They want to release something, therefore small perforations takes place. And then sperm plasma membrane and external acrosomal membrane outside sperm paper. There is some point fusions take place. And the acrosome reaction is associated with release of acrosomal enzymes that facilitate fertilization. It dissolves the egg membrane. In the sperm, you have a sperm membrane, you have the acrosomal membrane over here. Some points, fusion হয়ে যায়. Fusion হলেই, if there is a fusion, there are small perforations and these holes from which these acrosomal enzymes can come out. So this line I am talking, which happens only in the sperm. এই যে স্পার্মের মুখটা দেখলে, স্পার্ম প্লাজমা মেমব্রেন, অ্যাক্রোসোমাল মেমব্রেন, there is fusion. Fusion মানে কি হচ্ছে, এরকম একটা করে টিউব তৈরি হয়ে যাচ্ছে. So this is the sperm plasma membrane and this is the acrosomal membrane. So this fusion and this perforation is formed and things come out from inside the acrosome. So these are the process of sperm. So the process of acrosomal reaction, same thing, a sperm bind করলো, binding of the egg to the egg jelly coat, that is the corona radiata, sir. And enzymes released from the acrosomes. Enzymes digest the egg jelly coat. Sperm nucleus enters the egg cell. This is the acrosomal reaction. And there are some glycoproteins involved. Glycoproteins means carbohydrate plus protein is glycoprotein. And so one of those most important is this ZP3, which you have seen for this acrosomal reaction to take place. So then the next part is binding of the sperm to extracellular envelopes. After the acrosomal reaction, passage on these following enzymes: hyaluronidase, tubal mucosal enzymes. This is important for the sperm to pass inside the egg. Flagella action also helps. Then these are the acrosomal enzymes: esterases, acrosin, neuraminases. These cause lysis of the zona pellucida. You should remember these names with the enzymes. Once sperm penetrates zona pellucida, the zonal reaction occurs. Zona reaction occurs. This makes zona pellucida permeable to other sperms. Once one sperm enters, others are shunned out. So there is a block to polyspermy, prevention of polyspermy. So this is an important thing. This question comes: what is prevention of polyspermy? So there is one thing known as a fast block. Another thing is the slow block of polyspermy. The fast block is there is a change in the electric potential of the egg membrane. High amounts of sodium is pumped outside, and that will prevent all the other sperms, repel all the sperms. There is sperm ঢুকলো, lots of sodium is pumped out, fast block. Then and there activity. But then cortical reaction takes place, another a slow block will take place. Fast block কিছুক্ষণ বাদেই আবার ব্লকটা চলে যাবে, প্রথম শটই আটকে দিল. Then a permanent block is needed. You need a slow shot. So the cortical reaction, it occurs in fertilization when a sperm cell unites with the egg's plasma membrane, and that is the zona reaction. This reaction leads to a modification of the zona pellucida that blocks polyspermy. That many sperms cannot fertilize that egg. That is slow block of polyspermy. What happens over there? Water is absorbed, causing the membrane to swell and push the vitelline membrane away from the plasma membrane. You have the plasma membrane and the vitelline membrane in the egg. Lots of water comes and this swells. They are pushed away from each other. That means from the egg membrane, the sperms other sperms become more far away from each other. And this is the main thing which happens. They are part of the slow block to polyspermy. Now what happens? Let us see. Sperm binding and fusion to egg cell membrane. Sodium influx, membrane potential change, fast block হয়ে গেল. অনেক সোডিয়াম ঢুকে পড়লো ভেতরে. Then you have kinase stimulation. This is the slow blockage যাচ্ছে. Signal transduction test that will activate phospholipase C. That will activate inositol for triphosphate IP3 production and DAG or diacylglycerol production. This is a calcium signal transduction pathway. You have phospholipase C being activated, a kinase C, protein kinase C will activate phospholipase C. This will break down PIP to a 3 molecule, phosphatidyl inositol triphosphate. It will be broken down into IP3 plus diacylglycerol. This is the steps of signal transduction. This results in lots of release of calcium from the endoplasmic reticulum. And these lead to degradation of cyclins, MAP kinase, and cell division, inactivation of NAD kinase, conversion of NAD to NAD plus, all these cortical granule exocytes, these things takes place. Then there is a increase in intracellular acid, alkali হয়ে যাবে, and this results in restoration of the mitotic cycle, membrane biosynthesis, slow block to polyspermy, formation of hyaline layer, stimulation of protein DNA synthesis. My eggs, the oocytes are frozen in mitosis right from the when it's an embryo. Now when these cyclins and MAP kinase, they are responsible in cell divisions. They block. The block is removed and the cell cycle starts. So other sperms are the cell cycle has started. Now the sperms cannot enter anymore because after fusion, mitosis, the meiosis completes. So it doesn't now mitosis that zygote will start to divide. Membrane biodiesel, new membrane will form, and hyaline layer will form, and all these things will start to form, and these are the slow block to polysperm. Then you have the passage through extracellular envelopes. First is fusion of plasma membrane of oocyte and sperm. একই জিনিস পড়াচ্ছি. Head and tail of a sperm enter the cytoplasm of the oocyte, but the sperm plasma membrane remains behind. Dekhle sperm plasma membrane, only the nucleus enters. And the second meiotic, very important. Second meiotic division, it is frozen in the first meiotic division, right from the embryo. It is completed. The second oocyte was arrested in the metaphase of the second meiotic division, but now forms the mature ovum and other polar body. ওটা পরে oogenesis, oocyte cell division. And the fifth step, that is the fusion of the sperm and egg pronuclei takes place. Chromosomal material of the sperm decondensates and enlarges. Ovum, these things then still haploid. In the egg, it is haploid. Sperm is haploid. They fuse and form 46 diploid. These things, the pronuclei, they fuse. And this is once more the picture. A sperm is entering. The sperm is entering. This is the corona radiata, this region outside. Below that is the zona pellucida. Now this is the vitelline membrane. এই লাইনটা, এটা vitelline membrane. কোথায় দিয়েছি লিখে দিই. This is the vitelline membrane. And this the region space is the provitelline space. Perivitelline space, but surrounding the vitelline membrane. The egg nucleus, it is frozen in metaphase 2 of meiosis. Sperm enters, cortical reactions take place, the cortical granules, they allow it to enter inside, and gradually the second meiotic division is completed. Male pronucleus আসছে, this is the egg pronucleus, and they start polar body starts to take place. When this becomes actually divides into two, so the problem is one divides into one big and one small. This small is known as polar body. It will not. It's not. This will be the real egg. The small will degenerate. This in the next stage, this will also small. They will form two smaller polar bodies, and this will remain big only. They will die. কি 2n হয়ে গেছে, মাইটোসিস হচ্ছে না পোলার বডিতে যায় না. Twins are two types. One, you have dizygotic twins. One is the monozygotic twins. So once the zygote has formed, suddenly because of certain reaction, one cell becomes two cell and the two cells separate, and implant in two regions, it becomes twins. They were supposed to form 1%, it became 2%. Dizygote twins, for some reason, in that cycle, the female had made 2 eggs. So there are so many sperms, millions of sperms. So both of them get fertilized. So basically, they could have been at a different time. They could have been different individual, but but it happens that they are basically two different people forming, two different sperms fertilizing two different eggs. They were supposed monozygotic. They were supposed to form 1%. Twins formed because of that, not for polar bodies. And here meiosis 2 completes, and this particular zygote forms. The polar bodies are seen. It will go away after some time, but initially, if you see after fertilization, you can see these polar bodies are there. Sometimes you will catch one, sometimes you catch two. Okay, clear? Well, I'm stopping the sharing for those who are actually online. So, any questions? Online people, any questions? Ruchi? Nafisa?
No, sir. No, sir. No, sir.
No, sir. Okay. Nafisa, কিছু বলতে যাচ্ছ?
Sir, if the question comes about the fertilization, the process of fertilization, then it will be a long answer. If this will be the whole.
Can you repeat it once more?
If the question comes about how the fertilization takes place.
এক সেকেন্ড গুঞ্জা একটু বন্ধ করো তো, শুনছি আমি। এক সেকেন্ড কলটা বন্ধ করো। রিপিট দ্য কোয়েশ্চেন ওয়ান্স মোর, রুচি।
If the question comes that how fertilization takes place and everything, then we have to explain the whole thing now. Then it will
Yes,
be
of course. All the 5 steps. All the 5 steps.
Then maximum marks for this question will be 5 or
Yeah,
10.
it will not come like that. They will come broken. Gunya করতে পার, কোন অসুবিধা নেই। It will come. It will come. It won't come full. They will ask you like block 2 polyspermy. What is acrosomal reaction? You will never get a full question.
Oh, then it's okay. Thank you, sir.
You will get a broken up question and some questions which will make you think.
Sir, please send that book PDF, na?
Book Gilbert.
That book PDF, if you have, please send it.
Yes, PDF is available easily. You एक दिन कॉलेज में अपना पेन ड्राइव लाना, दे दूंगा.
Oh, thank you, sir. हम Friday को ले आएंगे.
Okay, then I close the class today.
Okay, sir.
ঠিক আছে, অনলাইন যারা আছে ক্লাস বন্ধ করছি।
ঠিক আছে, স্যার।
Okay.